Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Terus berpacu dalam keunggulan, lincah pembelajar untuk kehidupan. Inilah semangat baru, tekad raih kesempatan, wujudkan kampus terbaik selamanya.
Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah.
Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian.
Assalamualaikum. Good afternoon, Pak Dekan.
Assalamualaikum. Ini
Selamat
sinyalnya.
Kita mulai, Pak ya.
Suara saya kedengaran enggak ya?
Jelas, Pak.
Oke. Oke.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to the General Lecture for Summer Course 2023, presented by Department of Marine Science and Technology, Faculty of Fisheries and Marine Science, IPB University. We would like to express our heartfelt gratitude to the Dean of the Faculty of Fisheries and Marine Science, Professor Dr. Ferdinand Yulianda, Honorable Summer Course Coordinator, Dr. Beginner Subhan, the honorable speakers, Dr. Eric Horstman and Dr. Davin Setiamarga, honorable lecturers and staff of the Faculty of Fisheries and Marine Science, IPB University, and last, honorable ladies and gentlemen, the participants of the lectures. First of all, I give praise and gratitude to God who has been giving us blessing so that we can gather in this lecture without any obstacles. Today is a great honor for me, Niken Octaviandini, to be able to join and welcome each and every one of you in this opening of Marine Tropical Ecology Summer Course 2023. Please give your reactions. Before we get into the main event, there will be foreword and introductions which will be delivered by the coordinator of the Marine Tropical Ecology Summer Course 2023. I cordially invite Dr. Beginner Subhan to give a speech. For Dr. Begin, the time is yours.
Thank you very much, Niken. Professor Ferdinand Yulianda, Dean Faculty of Fisheries and Marine Science. Dr. Eric Horstman as speakers today, and Dr. Davin Setiamarga. And ladies and gentlemen, I'm very happy that we will kick off our summer course. The title of the summer course is Marine Tropical Ecology. And then this is the part of our collaboration, yeah, between Wakayama Institute of Technology, Japan, and also Twente, University of Twente. Thank you, Dr. Eric. And for this summer course, we have some participants from Malaysia, France, yeah, Japan, and also, yeah, mostly from Indonesia. And then the activity of the one course is a hybrid, yeah. So we will have field visitation, yeah, in next 29 of September, and we also conduct the online course. So this course, this summer course will be we can we can convert, yeah, become to SKS, yeah, for the students. So I hope all the participants can enjoy and follow the summer course and have a great experience. Yeah, because we have an international interaction. Yeah, between the international student and Indonesian student, and also from Indonesia there is not only from IPB University, Pak Din, Dr. Eric. So we have from other university. So quite varied of the student participant, yeah, for this summer course. Okay, thank you very much and enjoy the summer course. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon.
Thank you for the forward. Please give your reactions to Dr. Beginner Subhan. Next, there will be opening speech from the Dean of the Faculty of Fisheries and Marine Science, Professor Dr. Ferdinand Yulianda, that also marks of the official opening of the summer course 2023. For Professor Ferdinand, the time is yours.
Okay, thank you ya, Niken. Assalamualaikum warahmatullahi wabarakatuh. And good afternoon, ladies and gentlemen. Distinguished participant, yeah, also the Secretary of the Department of Marine Science and Technology, Dr. Begin, representative of department. First of all, let us thanks to the Almighty God, yeah, because His blessing we are able to join in the general lecture. You know. So welcome to the general lecture of the Marine Tropical Ecology Summer course, yeah, with the topic of expanding horizon, Indonesia Marine Tropical Ecology, and the ocean resilience on small islands. The general lecture is a series of summer course activities that will be held during 25 days from 26th of September to 20 October 2023. The summer course program was attended by 40 participants online and in field activities from Japan, Myanmar, France, Malaysia, and of course from Indonesia. The summer course activities will also be complemented by field studies. The field activity will be held on 29 and 30 September 2023 on Pramuka Island. This the small island of the Kepulauan Seribu. The online activities include the general lecture and online modules. Can be equal to credits. That's mean one credit can be equal to 45 hours for activities. Today we will listen to a public lecture from two experts who have experience from the Netherlands and also Japan. I say thanks to Dr. Eric Osman from Twente University, Netherlands, and also the David Setiamarga, PhD, from National Institute of Technology, Wakayama, Japan. So we know the Marine Tropical Ecology summer course in this time with the topic expanding horizon, Indonesian marine tropical ecology and the ocean resilience on small island. Involves the collaboration between various parties from various countries. The aim of understanding tropical marine ecology and rehabilitating coastal ecosystem on small island. The summer course is carried out hybrid, with 2 activities which are online on course and field activities. One of the series of activities in the summer course is the general lecture, which is held twice as the opening and closing the summer course series. The summer course we start with the opening general lecture, which presents a speaker from abroad, Dr. Eric and Dr. David, who will share the experience and insight regarding tropical sea and small island issues. In the opening general lecture I had today, to discuss marine tropical ecology and the ocean resilience on small islands. At the end of the summer course program, the closing of the summer course will be held simultaneously with the closing general lecture at the end of the series by presenting resources person who are expert in the field both from within and outside the country. The names will be announced by community. I'd like to say that. So we will wait for the closing agenda for the general lecture next month on October. For today, please enjoy the public lecture. Assalamualaikum warahmatullahi wabarakatuh.
Thank you for the speech. Please give your reactions for Professor Dr. Ferdinand Yulianda. Okay, so before we move to our main event, let's us take some picture for documentations. I kindly ask all the participants to open your camera and prepare your best pose. I'll guide the photo sessions by counting down from 3 to 1. There will be 2 pictures. Okay. We will wait for everyone to open the camera. Okay, we will start to take the pictures. For the first slide, 3, 2, 1. Okay, for the next slide. 3. Okay, 3, 2, 1. Okay, I think that's all for the documentations. So let's move to our next agenda, which is our main event today, the lectures we have been waiting for. We are fortunate to have 2 outstanding speakers today. There will be 2 sessions, which the first lecture will be delivered by Dr. Eric Hortsman from Twente University, Netherlands. I kindly invite Dr. Eric. Can you hear me well?
Yes, I can hear you very well. Thank you, Dea.
Okay.
Okay,
so the time is yours, Doctor.
All right, I will share my screen. Let me just check. I take the right one. I think you should be able to see my screen now. It works, right? All right. Well, good afternoon, everybody. Thank you for this opportunity, Dea, for inviting me to present in this summer course. And yeah, this first lecture, I'm going to talk about mangroves. So it's maybe not a very typical topic for a marine ecology summer course, but I think it's definitely relevant because, of course, there's a lot of mangroves in coastal areas. and they do actually affect what is happening in the coastal waters as well. So I'll shed a bit of light in this lecture on these interactions between the mangroves and the coastal waters. And the title of this presentation is Mangrove Rescue, and that is the title of a project that we've been working on for a few years, which is focusing on the resilience of mangroves and how we can use that resilience to protect coastal urban areas and coastal environments in general. So that's also the link to small islands, like there's mangroves fringing these small islands, and they can aid the protection of these small islands as well. So I will come back to that later on. If there are any questions during my presentation, please just raise your hand or put them in the chat. I will keep an eye on that at the same time. Yeah, so just a very brief introduction about myself. So who am I? I'm Erik Horstman. I actually did my PhD on biophysical interactions in mangrove forests. So it started early on, my interest in mangrove ecosystems. And my PhD was a project between the University of Twente and the National University of Singapore. So I actually did a lot of fieldwork in the mangroves in Thailand for my PhD project already. And then after my PhD, I moved to New Zealand for a postdoc where I focused more on sediment dynamics in mangrove forests. So that's what I was doing here. We're actually monitoring fine-scale sediment dynamics in between these metaphor routes here. And after that project, I moved back to the University of Twente and became an assistant professor in ecosystem engineering. So that's basically what I'm doing now. My work focuses a lot on ecosystem engineering, so that goes beyond mangroves as well. It's also about salt marshes, sand dune systems, all these kind of natural coastal systems. And I combine field observations with more controlled flume studies and then numerical models. Yeah, so that's what you will see back in this presentation today as well. So a brief overview. What we'll be talking about, I'll start very briefly on the ecosystem services offered by mangrove forests, and then I'll dive into their contribution to wave attenuation and sediment trapping, because those are the key functionalities that add to coastal protection. And then I'll finish off with a note on mangrove resilience, so how they can last into the future and how we can manage mangrove forests to enhance this resilience and to be able to actually benefit from their plentiful ecosystem services. All right, so I assume that most of you will have seen or be aware of mangroves and mangrove ecosystem services, but just a short recap. They provide a lot of different ecosystem services. First of all, they're ecologically important habitats for wildlife. They're a nursery for many fish and shellfish and crustaceans as well. Because of that, they also are a source of food. Fish, prawns, crabs, you can find a lot in mangrove forests, and there's a lot of people actually, dependent on mangroves for their livelihoods. For instance, here in Trang in Thailand. And then, they're unfortunately also used for wood, firewood, construction materials. And they have an important cultural function as well. They're being used for recreation, inspirational environments, and they even have some spiritual value, for instance, for the Maori people, the indigenous people of New Zealand. On top of that, however, and that's the function that we're more interested in for this lecture, is the regulating functions of mangrove forests. So they don't only improve air and water quality, but they also sequester carbon. So that's what they call blue carbon at a pretty high rate. They're about 10 times more efficient in storing carbon than any terrestrial forests. But they also attenuate hydrodynamic energy, so they reduce the energy of the waves that are coming into the forest, and thereby they also allow the sediments to deposit in the forest here. So they provide a bit of a buffer between land and the sea, and that is both for what is coming from the sea towards the land, but also for sediments and nutrients coming from the land. So from a coral reef perspective, this filtering function is actually also quite important. But that filtering function we won't be talking about. We will focus on, well, firstly, the wave attenuation function of mangroves. So how can they contribute to coastal protection? So looking at a mangrove forest, this is a mangrove forest in Kantang district in Trang, Thailand. And we see these waves coming into the mangroves. So these are relatively short waves. That's what we call wind waves. They have periods of about up to 20 seconds. And back in my PhD, we monitored the wave height reduction in this forest. So this is the Kantang forest here in the Andaman coast of Thailand. Thailand here, the Andaman coast, Kantang, and the Palian area. And we had actually two transects, so one in Kantang and one in Palian here. And firstly, we measured the bed levels in these forests with respect to mean sea level. We measured the vegetation density, so we counted all the tree stems and tree roots, we measured their diameter, and we converted that into vegetation density. And then we also monitored the waves propagating through this forest. So we measured wave heights and wave periods. And what you can see is that at both transects, these wave heights decline substantially throughout the forest, both in Kantang and in Palian as well. And what we actually also noticed is that the higher waves, so, well, they were still pretty low for the periods that we monitored, but up to 10 centimeters, the higher waves, they lost more of their height, more of their energy through the mangrove forest than the lower waves. At the same time, however, we noticed that the wave periods generally increased through the forest, so from the seaward side to the landward side, and that has got to do with the fact that the longer period waves are not attenuated as much as the shorter period waves in mangrove forests. So anyway, what we took away from these data is that this wave height attenuation, we computed a wave attenuation rate from that. So that's the wave height difference over the distance into the forest in relation to the incoming wave height. And we plotted that against the vegetation density, and then we got a very good relation between an increasing vegetation density and an increasing wave attenuation rate. So the denser the vegetation, and in this case that meant like in the Rhizophora zone where you have these stilt root systems that are very dense, there's a lot more wave attenuation going on than in the Avicennia zone where you only have these like 10 to 15 centimeter tall pneumatophores. So that was our finding for these rather short and low wind waves. But then the question is, what happens if you look into longer waves? So for instance, tidal waves or storm surges that have periods of 12 hours up to several days and that have wavelength of kilometers up to hundreds of kilometers. So if we look at tides first, this is a study that we did in the Whitianga estuary in New Zealand. And we basically monitored the tidal water levels all along this creek system. That was about 1 kilometer long. There was mangroves on both sides. And we monitored how the water level inside the creek and in the mangroves responded to the water levels out at sea. And we measured how much the water level at the back was lower than at the front of the forest. So this reduction of the high water level, the tidal high water level along this creek varied a lot along this 1 kilometer creek system. And what we see in this plot is that right at the entrance of the creek, so that is even to the left of this photo, the tide was actually amplified. So the height of the tide increased. However, if you go into the forest, so deeper into the forest towards the back of this picture, we observed maximum reductions of the tidal amplitude. So the high water level was up to 45 centimeter per kilometer lower than at the front of the forest. And the same if you look inside the forest. So these numbers are for the area along the creek, so directly here. But if we looked into the forest, this reduction of the tide actually increased to up to 95 centimeters per kilometer locally. So that's a pretty steep gradient of your water level. However, when we converted that to a reduction rate for the full length of the system, so when we compared the water levels in the back of the forest to the front of the creek system, the net reduction was only about 9 centimeters per kilometer along the creek and about 27 centimeters per kilometer within the forest. Still, you can see quite a significant difference between the creek and the forest, and of course that is due to the fact that along the creek, this is kind of an easy conduit for the water to move into the forest. Whereas inside the forest, there's a lot of friction with the trees and the roots. So there you see the greater reduction of the maximum water level. So then the next step would be to look at a storm surge. So storm surges are even longer than tides. Tides typically have a period of 12.5 hours. Storm surges can last for a number of days. And we were sort of lucky and unfortunate enough to be measuring during a storm surge in the Firth of Thames. So the Firth of Thames is in New Zealand as well. It is a long bay with an opening towards the north. And when a storm comes in from the north, you get quite a big storm surge at the bottom of this bay. So that's exactly where we were measuring. And this storm surge coincided with a king tide in November 2016. And that created water levels of up to 2.4 meters. So you can see here, this is the profile of the forest, the seaside and the land side. And the water level was up to 2.4 meters. And it only happens about once every 10 years. That also meant that the boardwalk and the platforms that we built in this forest to actually do our measurements were not high enough. So all our equipment started floating, and yeah, we had a bit of issue with that. But we got pretty cool data out of that. And what you can see on this slide is the different types of mangrove vegetation along the transect. So in the back, the dark green circle, we have pretty low sort of dwarfed mangroves. They're up to a meter tall, very dense though. Then further towards the front, they get taller, like about 3 meters tall, still pretty dense, but nothing like the fully grown trees that you would see in the tropics. And then at the mangrove fringe, the trees are a bit sparser, and right out on the mudflat, there's only a few individual trees. So what we measured here were the water levels at all these different diamonds that you see along this transect. And in these plots, you can see these water levels. So over a period of tides, the water levels were raised by this storm surge. And what we looked at was actually the difference between the water level at the front of the forest, so at the seaward side, and at the back of the forest. And for flood tides, we noticed that whilst the water level was going up, so from the blue line to the greenish line here and the black line on high water, we noticed that there was a very big water level difference between the front of the forest and the back of the forest of up to 50 centimeters. However, if we then looked at the following ebb tide, so going from this black line towards the green line and down to the blue line, so the water levels in front of the forest are dropping. But at the same time, the water levels in the back of the forest were not quite dropping yet. They kept increasing for a while. And that's due to the slowness of the system and the response of the system that is delayed compared to the water level, the speed of the dropping of the water level at the front of the forest. So in the end, the water levels at the back of the forest were only maximum 19 centimeters lower than the maximum water levels at the front of the forest, instead of those 50 centimeters that we noticed right on flood tide. Again, however, this means, so these 19 centimeters over 800 meters of mangroves come down to about 24 centimeters per kilometer of damping, of attenuation of the maximum high water level. And this is still quite a bit higher than the US Army Corps of Engineers rule of thumb for salt marshes, which applies a value of 7 centimeters per kilometer. And of course, that makes sense because salt marshes only have these relatively low grasses that are flexible, whereas mangroves have taller trees that are not flexible, so they provide a lot more friction to these storm surges. Yeah, so this effect that we measured in the Firth of Thames, that was put into a model by my colleague John Montgomery, and this analytical model actually computed the net reduction of storm surges maximum water levels over a mangrove forest in relation to its relative decay length scale. And this decay length scale is quite a complicated length of the forest, but what it basically combines is information about the vegetation density, the actual wetland width, the flood duration, the peak water level, and the shape of your surge. So for the Firth of Thames area, this decay length scale was about 812 meters. That means that is about the same as the width of the forest. That means that the maximum water level only goes down to about 80%. If the forest width would have been twice as long, so this factor would have been 2, then you could have reduced the maximum water level down to about 30%. So that may mean a great increase in the effectiveness of the forest for wave attenuation, for storm surge attenuation. Well, the impact of all these different factors we summarized in a review paper that was published earlier this year by Stijn Temmerman, myself, and a few co-authors. And here we really focused on all these different effects. So the wave period, the wave height, the effect of currents and water depth on this wave attenuation, the reduction of the wave height through the forest. And then later on, we related this to expansion and erosion of the mangroves, both laterally and from the top, but I'll come back to that a little bit later. What you see here, however, is also the relevance of the vegetation properties. So how stiff the vegetation is, the frontal area, the plant height. And in this paper, we specifically focused on that because we were comparing these salt marshes to mangrove systems. All right, the light turns off here. I've been sitting still for too long. So this brings me to the next part of this presentation, which is more about the sediment trapping effect of mangrove forests. Because the mangroves can attenuate, so they reduce the energy of currents and waves, and they can also thereby reduce turbulence. And that means that sediments that are transported into the mangroves can actually slowly settle. So they sink to the bed, they deposit on the bed, and they get stuck there. At the same time, often in these environments, we actually find cohesive sediments. So that's like clays and silts, and they can form flocks when the water is very quiet, when there's not much turbulence, not many waves and currents going on. And these flocks can actually get quite large, and that increases the settling of the sediments. So you can imagine that between these pneumatophores, for instance, there's not much going on in terms of water movement. And also, if you look at these dense root systems of Rhizophora trees, that significantly reduces currents and wave heights, wave energy. So that allows for sediments to deposit. At the same time, locally, this can increase turbulence because the water has to flow around these roots, right? And that can cause some erosive patterns, but that we won't be focusing on in this presentation. What I do want to show, though, is that here we measured the sediment deposition across a mangrove fringe. So a mangrove fringe typically starts with like some roots, some pneumatophores in front of the forest. Then you have the first trees, and then you have the forest. So we measured the sediment dynamics at these fringes in a number of estuaries, but here we see some results from the Whangapoa estuary in New Zealand and the Palian estuary in Thailand. And we see in these deposition rates that on the mudflat in front of the mangroves, there is some deposition already, but this deposition rapidly increases in this pneumatophore zone, so right at the front of the forest. And then inside the forest, it reduces again. So there's two trends there. The first is the increase of the deposition in these pneumatophores, and that is because this vegetation here, these roots here, actually reduce the hydrodynamics near the bed, and they allow the sediments to settle, to stick to the bed, and they won't be eroded again. But then because there is this increased deposition in this pneumatophore zone, then we see a reduced deposition further into the forest, just because the supply of sediments coming from the sea or coming from the river is reduced inside the forest. So these reduced dynamics between this vegetation here really allow for these sediments to settle and to increase the elevation of the forest floor locally. Well, apart from the mineral sediments, there's of course also organic sedimentation going on in mangroves. There's a lot of root growth in the soil. This is a cross section of a piece of mangrove soil. You see a lot of roots. There's a lot of leaf litter accumulating on top as well. So there's a high fraction of organic material, benthic production. So these soils are highly organic, increasing the volume of the soil, but also the roots actually increase the strength of the soil. So if water flows over the bed here, there's reduced erosion because the roots kind of keep the sediments together. And over time, this deposition can actually cause an increase of the bed level. So the bed level goes up and the mangroves can start colonizing this higher mud flat, and well, that's when you see a seaward expansion of the mangrove vegetation, like these seedlings here in this picture in I believe it was Malaysia actually. Nevertheless, oh yeah, so I will show an example of that actually, because we have, so this is the Firth of Thames that I showed before, where we monitored the storm surge. And this area has been expanding quite rapidly, even though the mangrove trees are quite low, so they're only a few meters tall. Over time, this forest has expanded quite significantly. So if we go back to the 1950s, in these aerial images, we see a sandy beach. So it shows up quite wide, and there's a few mangroves here. If we then look 25 years later, in 1977, we suddenly see mangroves all along the bottom of the Firth of Thames here. And that is due to the fact that there's a lot of sediment input from these rivers, from the Hauraki Plains into the system here. And these sediments are accreting the coast, and they're providing an accommodation space for the mangroves. And then another 40 odd years later, this is the current situation of the Firth of Thames. So you see this very wide band of mangroves. And actually, what is even more impressive is that the bed level of these mangroves is about a meter higher than the bed level of the land, the agricultural land, behind the mangrove forest. So these mangroves form a natural buffer to protect this low-lying land from floods. Of course, there's a dike here as well. They built a dike to make sure that they can keep the water levels out of the Hauraki Plains, these agricultural plains. But yeah, so the mangroves have managed to keep up with, well, to gain an elevation well above sea level. So in this case, up to 1.5 meters above sea level, mean sea level. And that is where mangrove resilience comes in. So that is the next part of my presentation, because this mangrove resilience is really related to how they can contribute to our coastal protection over time. So we've seen already that the mangroves can protect against storm waves and storm surges and also erosion, and that they can trap sediments. However, if storms hit, part of this vegetation is damaged, you will get some tree dieback, the attenuation function of the mangroves is reduced for a while. But at the same time, storms can also push sediments, so this yellow layer in this case, into the mangrove forest. So that can help the bed levels to go up. At the same time, it can also erode part of the front of the forest, so it's a little bit of a balance. The question is, over time, whether these storm deposits and the storm erosion that occur, whether there's a net deficit or a net gain of sediments. So that's what we try to depict in this figure. Over time, if a system has sufficient sediment input, it will bounce back from these storm impacts, and it might even keep growing over time. Whereas if there's a deficit of sediments, after every storm, the system will will lose some of its capacity, some of its height, some of its wave attenuation capacity, and over time the system will actually deteriorate and disappear. So the question here is, what you can see coming already, we're facing sea level rise. So if the sea levels are going up, the question will be, can these mangroves over time, taking into account storm effects and everything, keep up with sea level rise? And that was one of the questions that we were addressing in this mangrove rescue project. So what we were facing was, well, there was a bunch of data available already from these rod surface elevation tables. There's people that have been collecting very interesting data in mangrove forests around the world, mapping or monitoring the surface elevation changes over periods of like half a year, but it's very labor intense and we don't get too much information of the actual interannual fluctuations of the bed level. So the responses to storms or anything, we just have like one data point every half year or every year when they return to the mangroves. And also we have data available from sediment traps, but that's just data that measures sediment deposition over a period of like a day or a few days up to a week. So basically what we didn't know is what happens in this in-between period with a duration of weeks to months. So what fluctuations happen with in the bed level, and we sort of need that information to then make predictions for the future, because if we don't know how the system responds to sudden storms, sudden changes in these periods, we can also not quite predict what is going to happen towards the future when sea levels rise and where storm intensities might change as well. So that is what brings us to the research into mangrove resilience. And for that, we really have to combine hydrodynamics and morphodynamics. So that is the wave attenuation function, the sediment accretion, in combination with the ecological processes. And for resilience purposes, we're mostly interested in these surface elevation changes, and then following on that, the forest structure changes. So what happens to the mangrove forest, because that then is important again for the future wave attenuation capacity. And that is what we called persistence in this review paper that my PhD student Rick Gijsman published 2 years ago. So this is where numerical models come into play. And what I show here is a very basic model that I made for my PhD. So that's a while ago already in the meantime. And what we did is in this mangrove forest again in Kantang in Thailand, actually this side here on the side of the river, we monitored the sediment dynamics throughout this whole mangrove area. And we tried to make a numerical model using the hydrodynamics that we measured and the sediment input into this system. And then we used this model to figure out what would happen to these sediment deposition patterns across this forest if, for instance, the sea level would rise, or if sediment inputs would decrease, or if the vegetation would be affected by storm effects. So what I show here in this little plot is that with a change of the sea level, so an increasing sea level is, well, towards the right of 0, with a 10 centimeter change of this mean sea level, we already notice a 20% change of the deposition. So that's the deposition on the vertical axis in these different zones of the forest. And generally, the deposition would increase because there's more water in the forest, but there's also a zone here with the red triangles, the levee at the front of the forest, where the deposition would actually start increasing quite immediately. So that's going to be a problem, because that would mean that that area couldn't keep up with this sea level rise. However, this model was only looking at actual sediment deposition, so the and the effect on hydrodynamics of these trees. So what we did in this model is we implemented the presence of these trees. We just converted them into sticks, and then the model could simulate what was happening with the hydrodynamics, how much friction these trees would produce, and how much that would reduce the hydrodynamics. However, we know that more is going on, and that is what my PhD student is working on right now. So apart from the actual drag effect of the trees, so that is the above ground part of the trees. He also incorporated the stabilizing effect of the roots on the bed, so how they reduce erosion, and also the effect of drying. So because these are intertidal areas, like every ebb tide, the mangrove mudflats are exposed to the sun, right? And that means that with that exposure, they start drying out and they become a lot more solid. And then when the tide comes in again, the erosion is reduced. So from his new model, his extended model, including these three different effects, we actually noticed that that effect of the drying here in the yellow bars is a lot larger than the effect of the above ground vegetation on the water flows and the impact that has on sediment deposition. So this made us think on like what is actually the contribution of the trees itself, and to what extent is the sediment dynamics that we observe in this forest, to what extent are they actually imposed by just the fact that this area is partly above mean sea level. But then, of course, this relative height of the forest floor is created by the mangrove. So there's like multiple links that we have not included everything in the model yet. So for instance, in this case, what is missing is the dynamics, the organic sediment deposition. So root growth, leaf litter adding to this deposition. So that is going to be the next step, and that is what we continue to work on. How can we actually incorporate ecological processes in these kind of more engineering type models, simulating hydrodynamics and morphology? So this is an example of what we're doing right now, and we're trying to incorporate seedling development into these models. So this is a very simple morphology, and you can see randomly these seedlings start to grow in the model, and based on the conditions, they can survive or they cannot survive. Well, that is how it's usually done until now. So trees just start growing randomly. But what we started to do is actually taking into account the windows of opportunity that were established by Torsten Balke, for instance, taking into account that this area needs to be dry, so there can't be water for a few days for the seedlings to establish. And then they're sensitive to shear, to stresses, so they won't actually grow unless there's not much tidal and wave action going on for a while. And we've noticed already that taking into account these windows of opportunity makes quite a difference to the establishment, the tree establishment patterns that you can see here. All right, so that brings me to the last step of my presentation. How do we use these data? And this is also where the link to small islands, I'm happy I put that in my presentation as well, comes in. So we'll start with a few examples of mismanagement or where mangroves are actually forming a problem. So this is a project I did back in 2012 already on the island of Curaçao in the Dutch Caribbean. And they have, in Willemstad, they have this tiny little patch of mangrove forest. But the problem was that it's not very well connected with the open sea anymore. So there's an old coral reef structure here, and there was only this tiny narrow waterway. And that caused a water quality problem inside the mangroves. So there was a lot of stagnant water, there were mosquitoes, and it was quite smelly. And one of the problems was that the hospital is located right across the street. Here you can see it better, right across the street from the mangrove forest. Of course, that is not quite what you want. So the problem here was the limited water exchange. So what I actually did for this little project is that I proposed some improvements to this water exchange, improvements to the water system, so there would actually be more water exchange, and the water level back in this backwater would go up and down more, so you get more fresh seawater coming in and less problems with smell and stagnant water attracting mosquitoes. So that took a long time, but in the end, an engineering firm actually took up these plans and elaborated further on them, and then they turned it into the whole area into a mangrove park. where people can now walk around, they can actually learn about mangroves, and at the same time they implemented some of these improvements to get the water quality up and to reduce the mosquito plague. Okay, another problem that we were facing in New Zealand is that in New Zealand the mangroves are expanding quite rapidly. That's what I showed you for the Firth of Thames before, but that's actually the case in all the estuaries. And people don't like that. Like, we would be happy if in many places if mangroves were expanding, but in New Zealand they see it as a nuisance. These mangroves take over the estuaries, they block fairways, they block the view of the people living on the coast. So what you see a lot there is that they're actually what they call managing the mangroves, which basically means that they're taking them out again where they extend. So what we did here is that we wrote this book chapter on the importance of expanding mangroves, the dynamics of these systems, and how they can contribute to coastal management in the long term. And in the end, we even appealed like a mangrove management bill that was put in place by some of the regional councils to be able to take out mangroves, and they modified the text in that bill so it would not become as easy to actually do mangrove management in these areas. And then a last example, going back to a small island in the Caribbean again, is from Bonaire. So what you see here is the Lac Bay system with mangroves all around. And then there is this lake, which is Agua de Lodo, a backwater lagoon, which used to be a mangrove forest as well. The problem here, however, is that due to the sediment input from the land, it's quite a dry island, and there's lots of grazing by goats. So sediments are entering this system when heavy rainfall occurs, and these sediments have actually clogged the creeks in these mangrove forests, which caused a reduced connection between the backwater and the ocean, and that caused the mangroves to drown and to die over time. So this is what the mangroves in the back of this Agua de Lodo area look like. They just have died. And Mangrove Maniacs is actually trying to restore this area. They're digging out former creeks. They're putting in a lot of effort, but the question was like, how effective is this? Because it is a lot of work, and they didn't do any hydrodynamic studies into what the effect of this creek restoration will be. So we went there early last year, and our master student actually set up a numerical model. We did a lot of measurements first, of course, in the field of the bed levels, the vegetation properties, the water levels, the flow velocities. And he made a numerical model also in Delft 3D, taking into account the drag effects of the vegetation. And he then computed the residence time of this backwater, so this area here in the back. And for the current situation, he figured out that the residence time of this backwater was about 4 days. So then the good thing of the model is you can play around, right? So he took out all the creeks, and then the residence time increased to 7 days, so it would even get worse. But of course, that was not the purpose. The purpose was to find out how we could reduce this residence time most effectively. And then he figured out that just deepening or widening the existing creeks wouldn't actually do too much. What we really needed was to extend one of the existing creeks, or even create this new creek that you can see here in this picture. And that would reduce the reference time from over 4 to less than 3 days. So that would actually allow an increase of the water quality in the lagoon, and also improve the water level fluctuations in the backwater, so mangroves could hopefully re-establish there. But that re-establishment of the vegetation is something that we haven't modeled yet. That's something that is still to come. All right, so in conclusion, what is really important for successful use of mangroves for coastal protection and for allowing their resilience is that we combine monitoring with modeling and adaptive management. So that's pretty much what we try to do in our research as well. We monitor, we collect a lot of field data, we try to incorporate it in models, and then we use these models to try to predict what is going to happen to these systems under certain scenarios for different conditions. And then we use these models as well to figure out if you can do something to actually increase this resilience. So for instance, you can put some protective structures in front. You can increase the space behind the mangroves for increasing the tidal exchange. And these adaptive management strategies can then help to increase the resilience of the mangroves in the long term. And that would then help for us to be safer, to create safer living environments behind these mangroves. And yeah, on my last slide, you can see an example of that. This is a picture from Arborek Island in Raja Ampat. And here they are actually planting mangroves right on the beach to prevent for beach erosion. You can also see a rubble mound structure here, but of course the mangroves look nicer and are also more adaptive over time. All right, this was my presentation. I would like to thank all my colleagues who have added and contributed to all the fieldwork, the modeling. There is a literature list on the last slide of my presentation. I can share that later on. But first, I would want to know if there's any questions. In the chat or any raised hands?
Okay, so there's one person, Andy, already raised his hands.
Yes. Am I permitted to speak?
Sure.
Okay, thank you. Thank you so very much. First of all, let me introduce myself. I'm Andy, but my friends call me Ramzi because my name is very long. I wouldn't delve further into that. And secondly, I would like to thank you, Mr. Eric, for your very informative presentation. It was really interesting to see the issues about mangrove adaptability in other places besides Indonesia, in like Thailand or so in Kurakan and especially New Zealand, where you said that there was like problems with the people there regarding the mangrove. Okay, going to my question, I have a question regarding the measuring methods of your data, wave attenuation data. So I would like to ask, is there any specific devices or methods that you use to gather all that data, like the wave attenuation from the creek into the forest, and perhaps like can we know how many people was involved in that project on the field, measuring all that data. Maybe that is all from me regarding my questions. Thank you.
Yeah, thank you, Andy. That's an interesting question. So typically these field campaigns are quite intense, but we're never with a lot of people. So when we collected those wave data in Thailand, for instance, we only had like 6 stations along this transect through the forest. And at each station back then we use microwave loggers. They're basically just pressure sensors that are self-contained. So it's this big metal container with a lot of batteries in it, and then a pressure sensor and a memory card, and they register a water pressure at well, you can set the interval, but usually every few seconds. These days, we actually have equipment that is a lot smaller. We now use RBR pressure sensors, RBR Solo or RBR Duo. They are just like, they're smaller than a drink bottle. They're really easy to deploy. And it can last actually for a few months in the field, depending on your settings. So you don't need too many people to deploy them, and you can leave them in the field for quite a while. And yeah, the settings just depend on what type of waves you want to monitor and how often. So you can set the interval of your measurements to like 5 minutes, but then collect like 200 samples at 10 seconds interval every or at like less than a second interval, sorry, for every measurement. Does that answer your question, or did you have a follow-up question?
Yes, that answers my question perfectly. Thank you, Mr. Eric. And maybe just one more question, another topic. I was really interested with your Delta FM model, the one that you showed about the mangrove growing using algorithms. I think I'm not really I don't really know about that yet right now, but I want to ask, is there how many parameters did you use to build that model exactly, and is there any chance of it being inaccurate? Like how many percentage is the accuracy of that model, and also can it be added with more parameters? Maybe that's my last question. Thank you.
Yeah, that's a very good question, actually, and that is exactly the problem with these models. So we're trying to incorporate more and more processes. So for now, we focus on the vegetation, and we found already that just incorporating the windows of opportunity as we know them is not giving the results that we want. So for instance, the windows of opportunity are very strict. They say like you need 3 days without water for a seedling to be able to get a root and to stabilize itself. And then you need critical bed shear stresses to stay below a certain threshold for the seedling to not be eroded. And what we saw in the model is that we apply these thresholds, we actually don't get any seedlings developing in an area where we know that there is seedlings developing. So what we then started doing is that, of course, these seedlings are not all the same, right? And these seeds, the propagules are not. So we started introducing some random variability in the properties of the propagules, and that actually allowed us to recreate what we're seeing in nature as well. So that is a perfect example of trying to incorporate a process based on the knowledge that we have, and then figuring out that it's not quite like that. On top of that, there's a lot of dynamics that are not incorporated in these models yet. We don't do any soil chemistry, so pollutions or things like that. This organic sediment deposits by leaf litter or increasing root growth, we have not incorporated at all yet. So there's a bunch of jobs ahead of us still to incorporate in these models. I don't know what type of models this will be, if that's going to be AI generated in the future, or if it's still going to be process-based models like the models that we're working on now. But yeah, there's definitely developments going on in that area.
Thank you, Mr. Eric. That answers my question.
Thank you. Interesting question. Any more questions? Yeah, Jafari raised a hand as well.
Okay, is my voice clear?
Yes, it is.
So I want to ask about the sedimentation. I remember in the slide just back then that I believe aquatic vegetation is also responsible for sedimentation.
Yeah.
Relatively, would you say aquatic vegetation is just as effective as mangroves, or are mangroves much better at this sedimentation, and why? Like how much waves can aquatic vegetation in total attenuate?
Yeah, that's a good question. So if you look at the wave attenuation in mangroves compared to aquatic vegetation, what is really important is the water level compared to the vegetation. And if you have mangroves, the waves are usually, the water surface is inside the mangroves, right? So the waves are hitting the mangrove trees, and that is where most friction occurs, and that is how you lose most of your wave energy. If you look at aquatic vegetation like seagrass or maybe even coral reefs, the vegetation is under the water level. So the wave dynamics reduce towards the bed, and the area where the aquatic vegetation or the coral reef affects or interacts with the waves is further away from the water level. So there's less friction and you lose less of the wave energy. So in that sense, mangroves are more effective because they are right at the water surface. And that also means that they're more effective at the sediment trapping in the end because you lose more of that energy.
Okay, that answers my question. Thank you.
Thank you. Any more questions? Yeah, Tida.
Hello, nice to meet you, sir. Yeah, my name is Dia Susan. Yeah, I just want to ask you, I'm from Myanmar, and in Myanmar we also have the mangrove, and in Myanmar there is a lot of deforestation. There is happened by the human, mostly by human, and they make the deforestation. They make so many plants cut down for firewood. So I just want to ask you, although the mangrove can grow quickly, but if there's any chance to make the treated purification for the mangrove to grow more quickly. Is there any chance or not?
If we can speed up mangrove growth,
Yeah.
well, the best solution is to not cut them. We know that because once the mangroves are cut, like you lose the sediment accretion function of the mangroves, and then you sort of lose the natural area where they can develop well. So if we want to speed up mangrove development, they often do a lot of planting, but we know that is barely ever very successful, and that is related to the fact that the conditions are not right. So that it's underwater too often, that the waves are too high, that the currents are too strong. So what is really important when you're planting mangroves or when you're trying to speed it up is that the conditions are right. So for instance, what you see sometimes is if they replant old shrimp farms, shrimp ponds, it can work well because it's a really quiet environment. There's no waves, but if the water level is too high constantly, then it still doesn't work. But then it's an easy solution because you can drop the water levels relatively easily if the water control structures are still around. If you're on the open coast like here, that's a bit more complicated. Of course, you can't control that, but you could put in like these bamboo fences or something at the front to at least reduce the wave height. They've been trying that out in Demak, for instance, with the Ecology project over the past few years.
Yes, thank you, sir.
Thank you. Any other questions? I don't think there's any more raised hands at the moment. No. Okay. I think then I might hand over to Dea again.
I think there's one more person who already raised hand.
Yeah.
Mr.
Okay, thank you very much, sir, for the occasion for me. I'm Abdul Aziz from Hairun University, East Indonesia, especially in North Molucas province. Yeah, so several months ago, me and my friend went to Morotai Island, and we did research over there. We see the mangrove over there in Morotai, and very we saw very unique characters of mangrove over there, because several of the mangrove over there they grow from they grow on the rock, above the rock. So my question is, if normally mangrove grow from the mud, so how is it possible? What is the explanation the how can mangrove can grow on the rock, yeah, above the rock. I mean, the different, there is no sediment over there, there is no sand, but they can, the root can grow inside the rock. Yeah, I mean, that is very unique. So maybe you have the explanation to make us understand more about the characteristic of mangrove, especially the roots of the mangrove. Okay, thank you. Sir.
Yes, thank you, Abdulaziz. That's an interesting question. I actually have seen some of these mangroves on rocks as well. Like in Singapore, you have some, and in the Caribbean, I've seen some as well. The thing with mangroves is that actually, so the reason why in some areas we don't have any normal trees, but we do see mangroves, is that mangroves can actually tolerate salt water, and normal trees do not. So if a normal tree is getting their roots in the salt water, they will die, whereas mangroves can actually survive that. So that's their competition advantage. But then there's differences in where they grow and how well they do. So for instance, here we see these mangroves on a sandy beach. They do grow. But the thing is they grow best in muddy environments. So that's also where we saw them growing quite and expanding quite rapidly in New Zealand. That is a muddy environment. Because that's just because there's different conditions in the soil. There's more nutrients usually that they can benefit from in their growth. On these sandy beaches, you see they grow as well, but they grow, well, potentially a bit slower. And then on the rocks, that is even, more stronger, this trend. So that they're struggling a bit more to grow. They're probably not as resilient at all. Like if a storm comes by with high waves, you get a lot of reflection of the waves from the rocks as well. The trees might get damaged, and the recovery of these trees would be a lot slower than in a muddy area where there's plenty of nutrients around, where it's easier for the seedlings to establish again. Does that answer your question?
Okay, thank you, sir, for your answer.
Thank you. More questions?
Okay, Mr. Eric, maybe there's no other questions from the participants. We can finish this the first sessions here. Thank you so much, Mr. Eric, for presenting an amazing materials about mangrove resilience. We're really looking forward to meet you here in Indonesia in the future.
Yes, I'm looking forward to meeting you guys as well. Thank you for this opportunity. It was really nice to give this presentation. I don't know if you want to share the presentation, but I can send you a PDF. And if there's any questions in the future, just send me an email. My email address is in the presentation as well. Happy to answer questions later on as well.
Okay, maybe you can share the materials for today's presentations to the committee of the summer course, and we can help to share it to the participants today. Yeah,
perfect. I will send it today. Yeah.
Okay. Once again, thank you so much for today's materials. Maybe we can move to the next sessions.
Thank you.
Okay, so maybe we can give a reaction, giving an applause for Mr. Eric. You can give a love reactions, maybe. Okay, thank you so much once again. Maybe we can move to the next sessions. We will enter the second sessions. In these sessions, we will also have 1 hour presentations delivered by Mr. Davin Setiamarga, Associate Professor from National Institute of Technology, Wakayama, Japan. I kindly invite Dr. Davin. Can you hear me well?
Yes. Okay, I'm going to share my slides. Can you see the slides?
Yes, very well.
Okay.
Okay. So, Dr. Davin, the time is yours.
Thank you very much. Can you hear me well?
Yes. Yes.
Very good. Okay, so I'm Darwin Hadrianto Kaputra Siamarga. I'm an associate professor of molecular biology at the National Institute of Technology, Wakaima College, which happens to be a sister institution of IPB University. Today I'm going to talk about how to use molecular biology methods to dissect natural history and biodiversity. So I'm going to talk about, this is going to be more general than, it's not going to be very specific research. So relax a bit, and if you got something you don't understand, you can raise your hands and ask. Okay, so these are the three parts. My talk are going to be divided into three parts. These are the parts. So I'm going to talk first about this. We all know about what natural history is. Natural history is studying, we basically study living organisms on this planet. And then we basically study each individual at the individual level, and sometimes at the ecosystem level. At the ecosystem level, of course, we study how the animals, how the individuals or the organisms interact with each other. But sometimes we also study them at the organism level, where we study how the animal itself, the function, the bodily function of the organisms, for example, or how the genes or the molecules inside the organism works in order to maintain sustain activities as a living organism. And of course, natural history is natural history needs the field of classification. Wait a minute, needs classification, taxonomy, and systematics. Usually we would go to the field and collect living organisms, and then we would try to compare, for example, like this. Classification would be included in natural history studies. For example, you would see that they have different different forms, different tubes, and so on and so forth, and then you would basically classify them as two different species, for example. And of course, the science of natural history is already very ancient, because basically classifying things is one of the natural instinct of a living organism. Of course, for example, I took all of these pictures of cave paintings, from online, you know, from various sites. But basically, as we can see, we have the basic instinct to differentiate which animals are dangerous, which organisms are beneficial to us, which we should eat, and which ones we should avoid, and which ones will eat us, you know, for example. So it's a basic natural instinct. And of course, as I mentioned earlier, the core of natural history research is to collect samples from the field, and of course, to also do direct observations in the field. And usually when you would like to study the organisms more at the organismic level, you would collect the samples, and then you would voucher, or you would preserve the sample in the lab, you know, as a repository of biodiversity inventory. For example, like this one, this is a sample being deposited in the Nature and Science Museum in Tokyo. While these are samples collected samples of gastropods being vouchered in my lab. But the problem with natural history research is we always got anthropogenic bias in biodiversity research and conservation. For example, we always see this kind of posters, you know, we have to protect these cute kitty cats because if, you know, like, we cannot abuse them, we cannot do anything, you know. But then the problem is I've never seen any poster like this. cockroaches are animals too. Why would you defend the animal rights of kittens but not cockroaches? They're all metazoans. So there is this bias, And then, of course, because of such bias, this is a paper by Trude in 2017. So the line here in the middle would represent the number of species, right? So one species, one report. But here, as you can see, like mammals here, you got way more reports on compared to the number of species. Meanwhile, like say maxillopods or gastropods here or arachnids, spiders, you have much less reports than the actual predicted number of diversity. So, but why? It's because today mentioned something like this. Discussions on biodiversity often only focus on a small subset of species. Why? Because they are more likely to raise funds or are considered ecologically more important than others. So a lot of people would study, for example, tuna, and a lot of people would study oysters, but they won't study fireworms, for example, because they're being considered not sexy and then not a gold mine, right? So there is a bias also here in biodiversity studies. And the second problem is that non-biologically guided efforts of biodiversity research and conservation. This is a very famous example. I took it from a textbook, basically. The African cheetah. So in the past, when people thought that cheetah is endangered, what they did was they collect a lot of cheetahs and put it in the zoo and bred them, and then they would like send cheetahs to each other, to one another, in order to increase the number of populations. But the problem is, by doing so, they ended up making the genetic background of all species very close to each other, making them not as genetically diverse as they should be. So if a cheetah is prone to certain disease, all cheetah would be prone to the same disease. As you know, because it will be, and then so at the end of the day, it was not successful. But of course, this happens because the effort was guided by, of course, good heart, trying to conserve species, but not scientifically guided. Genetics, not genetically guided. And then, of course, there are bias and skewed old practices in natural history research. For example, of course, I already mentioned about collecting samples and vouchering them. But what you have to realize is that those samples being taken out from the field and being vouchered in your labs or in the museums are actually being taken out from the gene pools. And then so it's not actually being conserved. Well, they're being conserved as pickles. For example, one of my, and a person I happen to know, for example, collected like 7,000 individuals of certain shellfish for his doctorate, and then used only about 1,200 individuals in the actual dissertation. But the problem is, of course, he collected 7,000 individuals and basically removing 7,000 individuals out from the gene pools. It's like a lot of individuals. So it's not conservation, sometimes. So there are some contradictory and non-sustainable practice of biodiversity research. Another thing is that, of course, everybody wants to do molecular biology, DNA, biotechnology. For example, this one, DNA barcoding. Everybody wants to do DNA barcoding. DNA barcoding. Of course, I also do that. That's my specialty, my specialization. So to identify, this is DNA barcoding, identification of organism samples by interrogating query samples against database registered DNA data of identified samples. The problem is, of course, this words. You have to interrogate a database registered DNA data of identified samples. And there's also another new method. Everybody loves this method, eDNA, environmental DNA. You don't have to collect organisms. You can only collect the environmental matrices of the living organisms, such as the soil, the water, and so on and so forth. Some people even collected the air, collected DNA floating in the air. And but of course, in order to identify the naked DNA in the environmental matrix, you have to integrate the sequence DNA, the data you got from the environment, to a database. The problem is, do you have a reliable database? Well, a lot of people are using GenBank, blasting their samples to GenBank basically. But the problem is, of course, BLAST and GenBank are not developed for you to identify species. It's originally developed for you to identify homologous proteins from different known organism. For example, if you're collecting a sequence data of a certain gene from chicken, you blast it and then it hits, say, the PAX6 gene of human, you could say, oh, that's the PAX6 gene of chicken. You're not, of course, sequencing PAX6 and then blast it, and then you think that your sample is a human sample. So, but of course, you can use the same tool provided enough reliable data are being registered on GenBank. Well, this is an example. This is my niece when she was like 3 years old. Now she's already 12 years old. So I don't want her to see my presentation. She would get upset, I think, for using her picture. But how? Yeah, for example, there is this funny looking living organisms. You do not know what kind of organism this is, and then you collect its DNA, and then you blast it to GenBank. So, is there any representative sequence of the correct taxon of this creature? If there is, then yeah, of course, it will be identified properly well. But on the other hand, if not, my niece will be identified as a chimp, the closest organism to human. If there is no chimp data, she will be, again, for example, if there is, she would even be identified as a gorilla. If there is no primate data, she would probably be identified as a mouse. So the problem is, of course, you have enough data or not. So the problem with eDNA or barcoding is that when you sequence something and then you think, oh, this is a new species. Is it really a new species, or is just because the database is not complete yet? You really have to be careful. Well, the paper was written in, when was it? In 2009, I think. It's like way back. This is from my, wait a second, sorry. I couldn't see my, yeah. In 2009, this is among the first of eDNA research, one of the first eDNA research. At that time, they mentioned that most of the sequences are not identifiable, and so therefore they suggested that we will need a dedicated database in order to further clarifying sequences from unknown species. The problem is, of course, this already like, what's going on here? How come I can, okay, already 15 years, but because of the taxonomic sampling bias, we still do not have an exhaustive reliable database yet. And of course, we have a vicious cycle here. You need conventional methods in order to collect samples, but the problem is you might oversample. And then you will need to molecular method, but the problem is the old samples are sometimes not usable. And then if you use non-invasive molecular method, you don't have the database, a reliable database to compare your sequence. And of course, when you're trying to build a new database in order to reliably identify your samples, you will need to collect samples from the environment. So it will go on and on and on. And so we need to break the cycle. So my suggestion for Indonesia would be like to do museomics research. Basically, we can use old museum samples and sequence them, and then make a database of the samples, both. So we have the actual vouchered samples of the individuals at the same time as the the data, the DNA data that you might that will be needed for genetic identification of future samples. And Indonesia needs to do more taxonomic exploration. For example, like here, we know in 2012, I think we found, or 2010, we found a new species of mammals And of course, we always find new species of insects and then birds, So we need to actually explore and understand what we have in Indonesia, not only well, including in marine environment. So my messages would be like Indonesia needs to be more active in doing basic natural history research in order to explore and catalog our own natural resources, and we need our own repositories and databases. And the second part, I'm going to talk about fundamental and basic research. So I always love to talk about this, because whenever I go back to Indonesia, people will ask me, how would you use your research for product development, or how do you apply the result of your research? To be honest, my honest answer is I don't really care. I would leave other researchers to pick up the result of my research for a future application. So I'm going to start my I'm showing you this slides. Well, the slides is that when I was in the US, I was told that at that time, like way back in like 15 years ago or something like that, I was told that the US was basically focused their scientific life science research on these 5 topics. First is disease research, so it's of catchy diseases, like cancer, etc. And then the second one is, of course, energy problems. So it's like, that's why this is biofuel problem. The third one is research on what was it, on food problems. So how you use biology research to increase food products. And of course, the fourth one would be catchy environmental problems. And the fifth one, of course, being the US, is biomilitary. So if you're trying to do basic research, it's a little bit difficult to get money. But it was like way back 15 years ago. Now the situation is completely different. But I would like to argue that basic research, something that fundamental research, some people would call it as blue sky research or Star Wars research, actually is very important, and you need it as the basis. It's called fundamental because it's the foundation of the actual future research development. For example, like this, I'm showing different pictures of various vertebrates. This was me last month in Semarang. So what happened is that if you try using their morphology and then you try to group them, you might ended up grouping these animals like this. Why it is like this? Well, you can see that the chin. Anyhow, so these are, for example, they look different. They look similar, so that you might group them together, although they're actually genetically they're different, of course. And of course, for myself, some people calls me, I look like Kung Fu Panda, and some people, and of course, biologically, I might even look like a chimp, But anyhow, you never know. So how do you do that? You try to find a neutral marker, morphology marker, or morphological marker, or molecular marker, and you try to build a phylogenetic tree, right? Showing the interrelationship among different animals, among different vertebrates. And then usually using this tree, you will be able to show their evolutionary relationships. And then based on that, you can map the evolution and the evolution of certain characters. For example, like this, so the jaw probably like evolved around here. And then like, for example, like here, of course, it's cartilage, and this is heart bone. So they all have cartilage. So the heart bone evolved around here. And here, for example, you can differentiate ray fin fishes and the low fin fishes. Low fin fishes, of course, led to us, the tetrapods, right? This is the molecular phylogenetic tree of all vertebrates. These are the reptiles, and inside the reptiles you have squamates containing Komodo dragons and what else? Lizards, house lizards, snakes. I hate snakes, although they taste good. And of course, this group called Archosauria. Archosauria containing birds, crocodiles, and turtles. Yeah, birds are actually reptiles. But how come? People would say, well, birds are aves, not or aves. They're not reptiles, But no, they're actually reptiles, genetically speaking, and also, of course, morphologically speaking, when you're using neutral morphological and molecular markers. They're included in the group called archosaurs. Here, archosaurs. So birds are reptiles. They're archosaurs. They're separated from the squamates, containing lizards and snakes. And what about dinosaurs? Dinosaurs are members of the archosaurs, and how do they relate to one another? Basically, so there was a group in the U.S. They tried to extract. They succeeded in extracting actually protein sequences from fossils, and then they sequenced the protein. I think it's the collagen protein of Tyrannosaurus, and then they found out that Tyrannosaurus are close to chicken and yeah. Anyhow, so basically it is all birds, yeah, right. And then so you have T-Rex here, and then you have chickens here, basically. And so, yeah, birds are chickens. Birds and chickens are dinosaurs, and dinosaurs are not extinct. It's not like birds birds evolve. Dinosaurs evolve and became birds. No, birds are dinosaurs, period, So you have birds, which is birds or aves or avian dinosaurs, and the non-avian dinosaurs like T-Rex. But then when you compare, I forgot to translate it to English. So when you compare T-Rex with chickens, you have T-Rex, have like banana-sized fangs, but chickens, of course, do not have are toothless. But what happened? So these are the genes responsible for the formation of tooth. When they check what happened in chickens basically lost the upstream gene BMP here, and also they lost MSX1, MSX2. So because the upstream gene, the switch genes, the genes needed to switch on the signaling pathways, so they basically lost their ability to form because the downstream genes were all gone. They're not working anymore. So when they tried to find out the genetic disease, first what they did is that they put the BMP4 protein of mouse and injected it into chicken cells, and then they ended up making the chicken cells producing MSX1. And then because they have MSX1, they ended up, of course, able to form teeth. So they were succeeded in making chicken with teeth. Ayam bergigi. I forgot to translate this one also. But then, basically chickens have the genetic pathway to form teeth, but some genes are inactive. The upstream genes are inactive, and therefore they lost their ability to form the teeth. So there was some genetic disease called hypodontia or oligodontia. There are some people are unable to form complete set of teeth. So what happened is that when they try to find out the genetic cause of the disease, they found out that these people basically lost the MSX1, MSX gene. They're mutated. Therefore, it's kind of like similar to chicken. So what I'm trying to say is that they got, of course, some hints from basic research, from T-Rex to chicken to tooth loss in chickens. In 2009, Osamu Shimomura, the late Osamu Shimomura sensei, got a Nobel Prize for finding GFP from jellyfishes. Then later, Marty Chalfie used this GFP as an in vivo marker in living organisms. And then later, Roger Chen did genetic engineering, changing some amino acid in order to change the color of the GFP. So you got RFP or BFP, blue fluorescent protein, red fluorescent protein, and so on and so forth. So, what I'm trying is that when Shimomura Sensei, I watched the documentary about Shimomura Sensei. Shimomura Sensei, of course, when he was trying to figure out what he tried to do was only to figure out why the jellyfishes glowed. He only wanted to know what's the reason behind it. He didn't care about the application side of his research later. Later on, Marty Chalfie wanted to figure out the cell differentiation, and then he think he could use genetic engineering in order to do his research. And then later on, Roger Chen, of course, well, if you have more colors, of course, it's easier to look into different type of cells, you know. So what? Well, Shimomura Sensei, when he did his basic natural history research, he didn't care about the application, but other people will pick up on his result for and develop it further for downstream, right? So now, of course, GFP is being used in various applicative fields such as agriculture, pharmacology, and medicine. In Indonesia, we are always being asked to increase TRL, technological readiness level. But then, of course, the research in my lab are all TRL 0. So, because they're all fundamental research, but we have to, again, remember the fundamental research is the foundation of research. And therefore, if you don't have the foundation in doing research, it'll just topple. Everybody would say downstreaming all the time. Yeah, but now nuhai dihilir kan lamun hulu nasaat, right? Okay, so I'm going to the last part of my talk. It's about the research examples in my lab. So what I'm doing in my lab is, this is the mitochondrial phylogenetics of octopuses. So basically, we collected full mitochondrial genomes, we sequenced full mitochondrial genomes from a lot of different species of cephalopods, and then we found out that octopuses are actually do not form monophyletic genus. So you can see here like octopus here and the octopus here, so they don't form one group. Whenever I'm in Japan, I would say, so the octopus you put in your takoyaki probably are not octopus, you know. But of course, when I'm talking to Indonesian, I found out the other day that when I went to that the takoyaki in Indonesia contains chocolates, durian pieces, and cheese, you know, so not necessarily octopuses, so they're not octopuses anyhow, you know. So probably won't translate as well as when I talk to the Japanese. And using the similar marker, what I did is I also did molecular clock analysis and trying to figure out when they diversify. So we found out that many of the important speciation events or diversification events in the cephalopods actually correlates well with the extinction with past geological, in the past, it's like in the geological past, when extinction happens. For example, like here, PT is, of course, the Permian Triassic extinction, where 92.5% of marine organisms went extinct. Well, after that, and then here, for example, the KPG extinction is when an asteroid or meteorite hit Earth and caused the dinosaurs to an extinct. So later on, we also try to look carefully if there is any other environmental events. It's very interesting that we also found many of the diversification event actually correlates with past global warmings and global coolings of our planets. So when it cools, many of the species with high metabolic rate would increase its number of species. But then when it's cold, usually it's the one with slower metabolic rate would increase the number of species. Well, we haven't finished analyzing this. This is actually like a new result. So we're still trying to write a paper out of this. The second example is that is this paper where I asked my students to analyze old museum samples using next generation sequencer and sequence full mitochondrial genome. So we sequenced old museum samples like 10 or 20 or even 30 years ago, and sequenced its full mitochondrial genomes and did the so here we were able to obtain, this is the full mitochondrial genomes of Ceramaster japonicus, and then we did phylogenetic analysis using the mitochondrial genome data. And this is the third research where I also used museum samples, old museum samples, where I so I collected I collected various samples of Ceramaster japonicus and pentagonicus and Ceramaster arcticus from various museums all over the world. And well, I got only like about, of course, 65 specimens, something like that, not a lot, but because they're deep sea. But then I sequenced about I sequenced two genetic markers, and then we found out that actually, you know, like here, like the blue ones are supposed to be Ceramaster japonicus, but they're not formed together, so they're not of the same species. Actually, they're very far. While here, Ceramaster patagonicus here in Japan are actually not Ceramaster patagonicus, but apparently the same group as Ceramaster japonicus, and not the same as the other Ceramaster patagonicus from Alaska, for example. So this kind of like show that sometimes morphological analysis could be erroneous, and sometimes we need genetic markers to straighten things up. And this is my one of my main research at present. I collected, I did transcriptomics, proteomics, and genomics, so multi-omics analysis, and collected molecular information about shell matrix proteins, and then did molecular evolutionary analysis in order to figure out if the shells of various mollusks are homologous and how they form, how they evolve, and so on and so forth. So one of the results, the first result we obtained, like being reported in these two papers, are that the shell of the Nautilus, which is actually a member of the cephalopods, are actually homologous even at the molecular level to the shells of the, oysters, for example. Yeah, these are the genes we identified, and to be homologous to, you know, like the oysters, so there are six proteins are homologous. And also besides that, I also did similar analysis on the shell of the Argonauts, which is actually supposedly evolved from shellless octopuses. So the ancestors of the octopuses had shells, and then they lost their shells, and then later in the Argonauts they reacquired the shells. So we would like to figure out if the shells they required are actually homologous or not. So we did genomic analysis and so on and so forth, and we found out that they're actually not homologous. So it's being reported by the New York Times, so and so on. Also the local newspaper in Japan. And this is the molecular taxonomy research I did in Japan, where we collected samples, of course, from the deep sea. And then my colleague at the Museum of National Museum of Wakayama did morphological analysis, and then my student and I did the molecular phylogenetic analysis, and then we figure out that this creature is actually a new species. This is also in a local newspaper in Japan. And this is the one that we're still writing. We're trying to submit it. Hopefully, we can submit it this month or next month. We collected samples of this shell, the turban snails, from all over coastal area of Japan, and then we analyzed its genetic diversity, and we found out that using molecular clock analysis, we found out that they probably diversified during the Ice Age. You see, when Japan was, because of the Ice Age, were connected to one another, and then therefore the population from this side, from Japan Sea side and from the Pacific Ocean side are basically disconnected, and then therefore there was no genetic connectivity. And then this is the research I'm doing together with some colleagues in Vietnam. So we collected gastropods from the fish market in Vietnam, and then we did DNA barcoding because, of course, it's very interesting because we did some of the gastropods are being identified morphologically, and then they're using knowledge from morphological knowledge or taxonomical knowledge from species being already identified either in Japan or other surrounding countries, but we do not have any data about Vietnam actually. And so what happened is that when I did using 6 different genes and then we analyzed them, we found out that some of the species are actually not the same species as the one being reported morphologically. So now we are we are trying to publish this one and submit it, and then this is we are revising the manuscript, and then hopefully there will be a follow-up study identifying some species being identified as novel as an undescribed species through this study. Well, this is connected to traceability and food safety in Vietnam. And then this is a collaboration that I haven't been able to finish up, the collaboration with the late Dr. Hawes Madupa, my friend, my very good friend. But unfortunately, he passed away last year. So we collected animals from the Banda Sea here. And again, the funny thing is, of course, there is no database at all. So we still, at that time, I mean, at present, we still do not know which species are actually indigenous and which species are actually invasive. But we did find some species blatantly invasive because we found some Australian and some Madagascar species, like genetically we were able to identify species not, of course, coming in from Indonesia. And we kind of suspected that those species came in through ballast water of big liner. This is the last of my research example. I did, again, mitochondrial phylogenomics of the Medaka species. When I did molecular clock analysis, we found out that the 2 populations of Medaka were actually separated when the Japanese islands, the Japanese archipelago was formed. 10 years later, my colleague Yamahira Sensei obtained a sample from India, the Indian Medaka. And then when we analyzed did the molecular clock analysis, we found out that the Indian Medaka is actually the ancestor to all of the other Medaka in Asia. So basically, when India was still part of, India broke away from Africa and then moved up and then hit the Asian continent, causing the Himalayan mountain. At that time, some of the species endemic to the Indian subcontinent spilled out to Asia and then radiated and then ended up in various places in Asia. This is also being reported in the local newspaper. I got several research grants from the Japanese government, from several companies, mostly all from Japan. This is my college. So this in the middle of nowhere. So you see, naon teh di kampung pisan gitu, di lembur gitu ya. This is my campus, my lab here. I'm sitting over here now. This is my office. This is my lab. Yeah, and this is the main building here. This is located right facing the Pacific Ocean. These are my students. I asked them to collect samples in the field in winter. So yeah. So thank you very much for listening. So do I have to I'm returning this back to the MC. Thank you.
All right, thank you so much, Dr. Delphine. Maybe if there is any question from the participants, you can use the raise hand features if you wanted to ask about Mr. Delphine's materials today. Once again, is there any day?
Hello, I have a question.
Yes, go ahead.
Dr. Davin Sensei, sehat?
Alhamdulillah, sehat.
This is Media. So I have a question that's very interesting material and lecture. I show from your presentation and research, you have several sample or data from another country. I mean, how you use the data from another country? How about the biological or diversity ethic or permission? Maybe you can give me knowledge about that.
Thank you. Oh, si Dea. Okay, so I got my samples mostly my research being done in Japan are conducted on species only obtained in Japan. So I don't use samples from outside Japan. On the other hand, for some of the species I used from Indonesia, for example, I collaborated with Indonesian researchers, which I, of course, obtained the samples legally. I mean, my collaborators got the samples legally and shared with me. On the other hand, the samples from other museums all over the world, for example, well, they are vouchered specimens, and then I was able to, of course, they can share my samples. They can share their samples legally also based on the fair sharing of samples. Because if they're vouchered, I can basically loan the samples. I can get the samples on loan, and I can do analysis on those samples, because that's one of the reasons why the samples are being deposited in the museums. For samples you do not want to share, you're not supposed to deposit it in the museum.
It's also possible to get the data from the gene bank, right? To compare the genetic data.
For gene bank, of course, yeah, it's you can download it for free. You know, I mean, it's easy to download everything for free. You can basically download it the whole data set on your own computer. You can do it very easily, you know. But of course, for not yet published data, it's not registered yet. So after I hopefully I'll be able to publish my papers and let them go. And of course, we do need to compare our data with data from other countries in order to see the genetic connectivities of the organisms we're looking at. I hope that answers.
Okay, maybe is there anyone that still wanted to ask the questions to Dr. Davin? Maybe if not, I wanted to ask one questions to Dr. Davin. We know that these methods, the DNA methods, is not really popular in Indonesia, right? And I don't know about outside of Indonesia, so that's why I wanted to ask you, how many people that you already collaborate doing this kind of research?
You mean in Indonesia?
And also outside.
I think the method is actually, as far as I know, probably I've got my own bias, but I think the method is pretty popular even in Indonesia, because it's not that difficult to master, but of course it's a little bit expensive. No, not really. Actually, it's pretty like popular in Indonesia also, because I've met a lot of people doing analysis on DNA barcoding and eDNA, for example, molecular phylogenetics. Also, I've collaborated with various researchers in various parts of Indonesia. For example, I mean, for example, I have collaborators in Surabaya, in Semarang, in other different places. Meanwhile, in other countries, well, I would say that I wouldn't say that they're not popular. They are probably popular, but not yet well mastered yet, probably, because it looks difficult. But once you get the hands of it, it's actually like very straightforward and easier than many other analytical methods, I think. I hope that this answers the question.
Yeah, maybe one more question from me.
Sure.
Can you explain? Maybe you can you tell me what's the positive side of this method, rather than using the conventional one for the biodiversity research?
Right. So again, many of the let me show you this. There you go. Many of the conventional methods would require you to actually collect the actual organisms from the environment, right? So that means you will have to kill the animals or to kill the organisms, move it out from the actual gene pool, right? And you have to dissect it, and you will start, measuring things and that. But however, with DNA, for example, with DNA barcoding, what you need to do is only to clip a small amount of tissue, and then you just, without endangering the life of the actual organisms. On the other hand, with eDNA, you can collect samples of the environmental matrices, and you don't have to actually collect the animals. But of course, they're being used for different purposes. And another thing is that sometimes when you measure and then when you looked into something, sometimes it could be subjective. And then so your result could be biased with your own perspectives. However, DNA is straightforward. Basically, you collect the DNA, you get the sequence, and you cannot basically change the data or interpret the sequence based on your own perspective. The sequence is there, and then what you need to do is only to compare them. So I think it's pretty straightforward and not completely objective, probably, but it's slightly more objective than the conventional methods. Hope that answers.
Yes, very well. Thank you for the answers. And maybe once again, I wanted to ask for the participants, if you wanted to ask the questions to Dr. Davin, you can use the raise hand features here. And I also wanted to remind all the participants that to fill the absent in the Zoom chat, we already share you the links for the absence for today's general lectures. Okay, maybe if there is another questions for Dr. Davin, we can finish the sessions in here. Once again, I say thank you so much for Dr. Davin for the very insightful knowledge that you already shared to us. Hopefully, it will be useful and beneficial for us in the futures. And we're also waiting for meeting you in the future in Indonesia also.
Or in Japan.
Or maybe in Japan also.
Thank you very much. Looking forward to meet you all.
Okay. Before we move to the next sessions, I want to thank both of our speakers today, Dr. Eric and Dr. Davin, for sharing valuable insights to us. For the next agenda, we will have an explanations for the summer course that will be delivered by Ms. Dea.
Hello, everyone. Good afternoon.
Hello, good afternoon.
Yeah, so again, thank you very much, Dr. Davin and Dr. Eric for coming and then giving a lecture for our summer course, general lecture. And here for the student, stay tuned because I will explain about the online module that you will get from the summer course. So this is the open courseware IPB University. For the online and offline participant, you also can reach the OCW. Yeah, this is the link, ocw.ipb.ac.id. And then for the participant who already register and then send the email, we will register your email so you can access the material from the link. This is the user and also the password that you will can enter the system. Maybe we can share this next after the explanation and also after you give us the information about your email. And after that, you can log in with the ID. Please, next. So here you can find our summer course, Marine Tropical Ecology summer course from the Faculty of Fisheries and Marine Science. And then you will see there are several modules that you have to finish. And inside of the module, we have a quiz. So after finishing the module, you can continue to the quiz. And we have time from today until 20th of October to finish the module. And then what already said before, we will have a closing lecture in 11th of October. There's also will be 2 lectures giving the material, same like today. And when you finish the module and also the quiz, you can get the certificate and can convert to the credit or SKS. So just make sure you register in the link and then fill your email. And after that, we can register your email to the OpenCourseWare IPB and finish your module. So when you finish, we can give the certificate and you can convert the certificate to credit. This is also can be for Merdeka Belajar Kampus Merdeka, as you know that in IPB we have MBKM. So maybe you also can convert or claim in your university in Japan, France, Myanmar, and also in Malaysia. Is there any question? If you have any question, you can raise your hand. And also we already provide you the WhatsApp group for communication. So if you have a question or difficulties about the module or about the online module, you can give a question in the group. If no question, yeah, just make sure again you send us the email in the link, yeah. So Sensei Davin or Eric, if your student want to join this online course, they still can join and then also fill the registration that we already shared to you. So please share this to your college or your student in your university. Thank you. So maybe if there is no question, I give again to the Niken, please Niken.
Okay, thank you so much, Ms. Dea, for the explanations. Maybe one more announcement that I wanted to share to you guys. So, in October, our department will also held the 5th International Conference on Marine Science 2023. You can access the website and for further informations on itk.ipb.ac.id ICMS 2023. Okay, you can find further informations about the event on the website, and the deadline for the abstract submissions is on 5th October. We will also send a link to the website in the chat room. All right, maybe that's all for the announcement. So ladies and gentlemen, those are the agendas we have presented to you today as we have finished our lectures. Once again, we would like to thank our speakers, Dr. Eric Horstman and Dr. Davin Setiamarga, for sharing a very insightful knowledge and valuable insight. Please give reactions to the speakers. All right, thank you so much. I also want to thank all the participants for the participations during these sessions. Hopefully, the presentations will be beneficial for us. From the deepest of our heart, we do apologize for our mistakes in presenting this event. Maybe that's all for today. Once again, thank you for making time in your busy schedules to join us here. I wish you all a pleasant day. We hope to see you again soon. Here I end our agenda for today. Wassalamualaikum warahmatullahi wabarakatuh. Thank you so much, Dr. Eric and Dr. Davin.
Thank you.
See you soon. Sensei.
Aduh. Tapi kok inian kecepatan baru yang ngomongnya teh orang teh.